first speaker that Stas Shvartsman. Now he's from Princeton University. He will talk about back to the future of systems genetics. And we really appreciate that Stas is with us because it's the middle of the night now in Princeton. So thank you very much, Stas. All yours.
Okay. Good morning, everyone. Can everyone hear me?
Can you hear?
No, not very
well.
Well, it's slow, but it's...
Exactly, that's what I said, yeah. Okay.
We could hear you very well.
Yeah, I know. Stas, can you maybe improve your microphone somehow?
Yeah.
We're still waiting some technician to come here to the auditorium to check the volume.
Okay. Can you hear what I'm saying now, or you can't hear at all? Hello?
Yeah, just a second.
שומעים אותך פה?
אותי הם שומעים מצוין, זה דרך הזום, זה אותו, הם שומעים מאוד חלש.
בשביל הזום אתה צריך להשתמש במדור, את זה תשים עליך.
אני צריך?
כן. כדי שישמעו אותך בזום.
לא, המרצה הוא בזום.
אותו לא שומעים.
אותו לא שומעים, לא אותי. אפשר לעשות בזום עכשיו?
יכול לדבר רגע?
Stas, can you say something so we can
Hello? Hello?
He's the tech guy.
אחד, שתיים. כן, אבל חלש.
Hello?
הווליום של המחשב עצמו. Can you talk again?
Yeah, hello. Can you hear me now?
Okay, great. We can hear you. Should we close the video or leave it open?
No, leave it open.
Okay. Should he start?
Yeah. Stas is all yours.
Okay.
We can hear you now.
Yes. Okay. Good morning. So I'm sorry I could not be there in person. If I could have traveled, it would have been my fourth conference, but I'll hopefully will make up for it next time. All right. So it is 2:30 in the morning here at Princeton. So if there are any glitches with the presentation, you can blame it all on the time difference. Okay, let me see. Second. All right. So can you see my slides now? It was not moving a second ago.
Yes.
Yeah. All right. So I will tell you today about a paper that was written almost 100 years ago. So the paper was written by a Russian scientist, zoologist, Sergei Четвериков. It was written in 1926. Четвериков did not write too many papers in his life. This paper was written in Russian. It is not very well known, but it had a lasting impact on biology in the 20th century, and I'll try to explain to you why I think this paper is very important. So the paper starts with the following introduction. It says that genetics is a great science, and you can imagine things that were happening 100 years ago. Mendel was rediscovered in the early 20th century. Morgan was showing that genes are located on chromosomes. Things were moving very swiftly. It was the youngest branch of biology. And yet the paper starts with the following sentence. It says, genetics is in a contradiction with conventional views of evolutionary concepts. And in this lies the reason that Mendelism was greeted with such hostility on the part of many outstanding evolutionists. The present article sets itself the goal of clarifying certain questions on evolution in connection with our current genetic concepts. So, what was the conflict between genetics and evolutionary theory? So, Mendel, if you read his paper, you will find out that he started at 8 years before the paper was eventually published in 1865. By the time that the paper was published, he certainly knew about the work of Darwin and Wallace. In fact, that if you look at the introduction of Mendel's paper, you will find that even though he was an extremely modest person, he writes that what he was doing is the only right way by which we can finally reach the solution of the question of the importance, which cannot be overestimated in connection to the history of evolution of organic forms. So here is an Augustine monk explicitly mentioning the evolution of organic forms right in the introduction to his paper. So he wrote it in 1865. The paper was also published in an obscure journal. And then the paper was promptly forgotten. But from the introduction of Mendel's paper, you see that he clearly knew about the fact of evolution. By the time that Mendel's paper was rediscovered, Mendel was dead, Darwin was dead, but Wallace, who is the co-founder of the theory of evolution together with Darwin, was still alive. And he was witnessing the rediscovery of Mendel's paper. He was witnessing the swift pace of work on heredity and evolution, which was the missing piece of his and Darwin's theory. And yet he felt necessary to comment on the current state of things. So this was one of Wallace's comments in 1908. Darwin did not altogether exclude discontinuous variations, a la Mendel, of the types when a trait has two distinct features, for example, the color of the pea or the roundedness of the pea, but he soon became convinced that these were quite unimportant. These curious phenomena, and by this he means discrete traits or discontinuous variations in nature, may be of interest as furnishing materials for the study of the theory of heredity, but they have absolutely nothing to do with the origin of the modification of species. So here is one of the two founding fathers of the theory of evolution explicitly saying that the kinds of traits that were described in Mendel's paper and that were demonstrated again and again in a number of different plant and animal systems has absolutely nothing to do with the progress of evolution, with the modification, and with the origin of species. So this was 1908, and the things did not get much better even a decade later when Morgan and his students in Columbia University established a firm link between cytology or cell biology and genetics, demonstrating that genes are located on chromosomes. It was not clear what genes were, but Morgan and his students in the fly room at Columbia demonstrated they are located as beads on a string on the chromosomes, and they behaved during their segregation in the reproduction process very much like the chromosomes did in the microscopic observations of cytologists. So there were several arguments on the part of evolution. One of them was that evolution has absolutely nothing to do with discontinuous variations. Second, it was not clear what genes were. And third, it was argued that mutants or monsters or freaks or sports that are studied by geneticists arise only as a consequence of selective pressures that are applied in artificial conditions of, let's say, of animal breeding practice or of laboratory procedures. So this was the conflict. It was first of all, it was said that the discontinuous variations are irrelevant, and then people said that mutants that are studied by geneticists are not found in nature. So the very critical experimental proof that essentially destroyed these objections was provided in two parts. First of all, in 1918, Fisher wrote a paper and that said that as a consequence of very simple math, essentially central limit theorem, continuous variations can be explained if you view a certain trait as a sum of effects of a relatively small number of genes, 5 or 10. And very quickly you start generating normal curves, Gaussians, that were studied by people who favored continuous variations over discontinuous traits a la Mendel. And then there was still an argument about whether mutations are found in nature, and it is this argument that demonstrated that mutations are found in nature, that they're found spontaneously, that was provided by Chitveryakov's paper. But let me first provide the historical context and the geography of where the work was done. So the work was done in Moscow in the Institute of Experimental Biology that was headed by Russian biologist Nikolai Koltsov. Koltsov was a classically trained zoologist. He traveled extensively before the Russian Revolution, and he met a lot of luminaries that visited biostations in Naples, and he knew a lot of the people who were the movers and shakers of cell biology and genetics in the beginning of the century. After the revolution, he and Koltsov was actually he opposed Tsarism. He left the Moscow University in protest over repressions by the monarchy against students and workers. And he established an Institute for Experimental Biology in 1916. And already in 1924, it was known that something very interesting is happening in this institute. He was invited to travel to the International Congress of Zoologists in America, and he delivered the following address. He actually could not travel there, but the address was published in Science, of all places. I found it just a couple of days ago, and it is very interesting to read what he writes in his address about his institute. And I have this quote here specifically for Ute. He says that there are many great places in which biology is done. So the laboratory of Jacques Loub pursues the application of physical chemistry to biology. Morgan's laboratory is devoted to genetics of Drosophila, but it would be very interesting to unite these two scientific disciplines in the same institution. In every science, the best results are to be obtained when the same theme is treated with two quite different methods belonging to two different scientific disciplines. Therefore, I have tried to put together in my Institute of Experimental Biology many different sections such as cytology, biochemistry, physiology, behavior, genetics, and eugenics. So this is an example of an interdisciplinary institute in the beginning of the 20th century in Soviet Russia. So if you read the description of the research program, then minus eugenics, you will find the kinds of things that were happening at the start of systems biology approximately 25 years ago. So this was the setting in which Chitveryakov was working. So from the same report, he describes what was actually happening in this institute. In particular, he writes that Professor Müller, so Müller was one of the three star students of Morgan in Columbia. There were Bridges, Sturtevant, and Müller that co-authored with him the book about the physical basis of heredity. So Koltsov writes that Müller visited Moscow in 1922, and he brought to us from America a very valuable present, 32 living races of Drosophila melanogaster. We have now published a Russian translation of Morgan's Physical Basis of Heredity. So the book is Morgan's book, but it is co-authored with three of his students that were, some of whom were not even 20 when they started working with Morgan, and here they were in the early 20s publishing this major book. Thus, we are able to study the genetics of this famous fly on a large scale. And this is a photograph with Morgan in the black suit and the shirt and black tie in the center, and microscope in Moscow with students and researchers in Koltsov's Institute. So this is the connection of Morgan's school and Koltsov's interdisciplinary institute. So Koltsov asked Четверяков, who was his, Четверяков at the time was approximately 40 years old. He was a junior colleague of Koltsov, who was a director of the institute, and he asked Четверяков to start a laboratory of genetics. Четверяков was trained as a classical zoologist. In fact, he was an expert in butterflies and moths. And he recruited a team of 10 young zoologists, all of them in their early 20s. They read Morgan's books and they started discussing the literature on genetics. And they had to catch up with what was happening in the world during the time that Russia was going through the period of two revolutions, bloody civil war, and as a consequence, did not have access to literature and information about what was happening in the West. So this is the photograph of a lot of the members, or some of the members of Четверяков's team. So here they're sitting at the steps of the biostation, which for many years later on was the biostation for the experimental summer practicum of the biology students in the Moscow University. Четверяков established a very special format in which they discussed current trends in genetics. This format was called Droz-Soor. So Droz comes from Drosophila, and Soor is an abbreviation of Russian terms, which means совместное орание, which means joint shouting. So it's a special research format of a research discussion that was invented just for this purpose. And in this discussion format, there was a senior person like Четверяков or Koltsov who guided the discussion. And all the participants could interrupt each other at any moment whenever they had something important to say. So of course they had to prepare for this discussion. And of course, when somebody interrupts you, you can be upset. So because of this, all members, all people participating in the discussion had to be on good terms with each other. And because of this, they had to vote by secret ballot of who can be admitted to this discussion. But this was a freewheeling discussion in which they tried to identify open problems of modern biology and define problems or projects for their future research. So in these discussions and these joint shoutings about the genetics of Drosophila, they focused on the gap between population, between evolutionary theory and genetics. So what was accomplished? So what was accomplished is a very clear answer to questions about whether mutations can arise in the wild and that they're not a consequence of artificial selection pressures. And if mutations arise in the wild, then what explains the rarity of mutants and monsters in natural populations? Because one of the arguments against the importance of mutants, so the word coined by De Vries, was that if you look at natural populations, then you see then you see these deformed forms of plants and animals extremely rarely. So they might be arising, but they have absolutely no consequence for the progress of evolution because they're immediately weeded out. So this is the summary of Четверяков's paper. It was a theoretical paper. Let me summarize things for you, and then I will tell you about the experimental proof that was provided by his students. So first of all, the paper starts by saying that there is absolutely no contradiction between Mendelism and Darwinism, and that mutations should not be viewed as artifacts of laboratory procedures. Then he said that these natural populations that are apparently quite homogeneous phenotypically are actually extremely heterogeneous in their genetic makeup. So Chitverikov used the word generation for mutation, and he said that natural populations are filled with variants of genes, and they are maintained mostly in a heterozygous state. So most of these mutations are recessive. And because of this, they are hidden from the eye in a phenotypically identical but genetically diverse individual. So this is a completely trivial statement by modern terms, but apparently this was not clear. And in fact, many people who disputed the presence of the gene, just like just a couple of decades before this discussion, many people, including the leading physicists like Mach, disputed the presence of atoms. They did not understand that genes are discrete entities, and they thought that upon mating, differences will be infinitely diluted, just like a drop of ink would be diluted upon mixing with a larger and larger quantity of fluids. Okay, so by using arguments that are based on a relatively small set of mathematical tools, such as Hardy-Weinberg equilibrium, he said that populations absorb the arising mutations like a sponge, and that selection acts on already existing gene variants and does not itself generate new mutations. And when a genotype or a variant is selected, it is selected in an organism that already carries a large number of variants that generate a genetic background or genetic milieu, like he said. So all of these things are completely standard and would have been not argued by any undergraduate in genetics, but this was the hottest controversy between the new field of genetics and Darwinism that was a largely descriptive science at the time. Okay. So what was the experimental proof? So the paper was written in 1926, but already a couple of years before that, Chitverikov's team started a large-scale population genetics experiment, the first of its kind, and we will see the modern versions of these experiments. They traveled about 1,000 miles south of Moscow to a place that is called Gelendzhik. So there were two members of Chitverikov's team traveled there, and they collected approximately 300 fertilized Drosophila females. They brought them by train to Moscow. They let them lay eggs and they collected the progeny, and then they mated the progeny among themselves. So they mated daughters and sons of the same fly and multiple such things with themselves, and then they mated the progeny of the progeny. So these were experiments that mimicked one-to-one Mendel's experiments with the peas. So they did not establish a true breeding lines, but they just examined the first generation and the second generation. So in the same way in which in Mendel's experiments, if you cross phenotypically identical plants, you will uncover segregating traits if they exist. They uncovered numerous Mendelian traits. I will show you a couple of examples now. They discovered that every one of the lines that is established from every one of the fertilized females harbors between 5 to 6 different mutations. And these are just morphological traits that can be observed by adults using the crude methods that were at their disposal 100 years ago. And these variants, they ranged from relatively small morphological modifications to major changes. So here are some examples. So this is a tally of mutations that they discovered. So for example, they found that one of the mutations was present in 20% of all lines that they established. So you could not have guessed that it is there because you were working with heterozygotes. Some of the mutations, they looked like radius incompletus. So if you're not working with Drosophila, I need to explain to you that you're looking at wings of insects. On the right, you have a wild type, a normal wing. You have veins that are straight lines that run longitudinally or transversely. And in some of the mutants, these veins are imperfect. So, for example, this vein is incomplete or interrupted. So, such a mutation was found in a substantial fraction of the population. So, it's a minor modification. So, this fly can fly and mate just as a normal fly. But what is absolutely remarkable is that in some of the flies, they found things that takes them seemingly to a different kind of species. So there was a mutation that was called an aristopedia. So this was a mutation that converts a fragment of an antenna of an insect to a leg. So this is an animal that is presumably on the way from becoming, so this is of course not what is happening, but it shows that a genetic modification can seemingly convert something with 6 legs to something with 8 legs, like a fly into a spider. Okay. So some of these mutations were lethal, and when present in homozygous state, did not give any progeny or gave dead animals. But what is important is that the mutations that they uncovered in this natural population, at least some of them, corresponded one to one, and this is something that they could prove using Miller's line, with the help of which they could map the mutations in their natural populations. They corresponded to mutations that were discovered by Morgan in the laboratory. So this was an experiment. So to prove or to get around this catch-22 situation, that in order to study mutations, you have to bring them in the laboratory. In the laboratory, there is ether, there are artificial conditions, there is artificial food. So here is something, and in 2 generations, and flies breed in 2 weeks, you can just uncover the presence of mutations. So many such examples were present in the wild population. So everything was going great. Четвериков wrote this paper in Russian. He presented it in 1927 in the Congress of Genetics in Berlin. But in 1929, he was arrested by Stalin's secret police. He was exiled from Moscow, and his laboratory was disbanded. He never returned to his studies in Drosophila and could not complete the work, including the English translation of his 1926 paper. When he was exiled, and he was exiled to Sverdlovsk, Yekaterinburg, which is the place, the historians of you know that this is the place where Tsar and his family were shot in 1918. He worked as a zoo consultant there, and then he taught in a vocational school that trained experts in pest control. He was banned from visiting Moscow, Leningrad, and other large cities. And in spite of this travel ban, even working in these remote places, he managed to discover and characterize a couple of new species of butterflies. Eventually, he returned to research in the late 30s. He worked in Gorky or Nizhny Novgorod, where he worked motivated by military applications in silkworm. And in 1948, during the time of Lysenko's purge, he was again fired and banished from science. So this remarkable trajectory was interrupted and the school was disbanded. Fortunately, one of the people that you saw in the photograph of students that at the steps of the summer experimental lab left Russia, and he went to Berlin in 1925 when he was 25 years old. So he went to the Institute of Oskar Vogt to Berlin in the Kaiser Wilhelm Institute for Brain Research. Vogt was a psychiatrist and neurologist that was invited to Moscow to help and supervise the dissection of Lenin's brain. He was interested in the genetic basis of psychiatric disorders, and he knew that genetics is something that he is missing in his scientific toolkit. And he asked Koltsov and Четвериков for help in identifying a young person that can start genetics efforts in Germany. So Timofey Rysovsky left in 1925 to Germany. He became one of the leading geneticists of the day, working on 3 research directions that he brought with him and successfully developed in Berlin. So these directions were on the genotype-phenotype connections, on the mechanism by which mutations actually arise, and in the genetic makeup of natural populations. And TR, as I will be abbreviating him in the rest of the talk, he brought the style of these joint shouting or draw sword discussions that he learned and absorbed like a sponge from Koltsov and Chetverikov to the West. In the early 30s, Timofey Rysovsky, so here you see him in a photograph in his laboratory in Berlin, met young Max Delbruck, who was looking for new problems in physics, and he turned to biology, strongly encouraged by Niels Bohr. And together with Timofey Rysovsky and who was a geneticist, and Carl Zimmer, who was a radiologist working on the dosimetry of X-rays, with which Timofey Rysovsky and Muller were generating mutations, they wrote a foundational paper in physical biology. So these relatively young people, the oldest author was 35 years old. Zimmer in 1935 was 24 years old, and Delbruck was in his late 20s. They wrote a paper on the nature of gene mutation and on the nature of gene structure at the time when absolutely nothing was known about the structure of the gene. So in this paper, they argued that the way in which radiation interacts with chromosomes and with nuclei and with the physical substance of cells suggests that genes have chemical structure, and that you can think about genes as or an assembly of genes in chromosomes as one large chemical molecule, one large molecule, which is one of the ideas that was proposed by Koltsov and that Timofey Rysovsky brought to the West. So the paper was also published in an obscure place. It was Proceedings of the Göttingen Scientific Society. And later on, Delbruck joked that by publishing the paper in this place, it was given a first-rate funeral. But a couple of years later, a popularized version of this paper was presented to a much broader audience by Erwin Schrödinger, one of the founders of quantum mechanics. So this paper was based on the set of lectures that was given by Schrödinger in Ireland. And this paper was what brought many physicists and many young people interested in the quantitative understanding of genetics and the material understanding of what genes are to biology. So here is a snippet of review of this paper. Let me try to move my thing here so that I can read. So here is a snippet of review of this paper by Haldane, who was one of the major architects of the modern synthesis, what eventually brought together genetics and evolutionary biology. So this is Haldane's review of Schrödinger's book in Nature. He said, "The book is the one to which one comes back again and again, and I have lent it to several genetic colleagues, and the verdict has been uniformly favorable. We may disagree with details or even with the fundamental principles, but we cannot stop reading it before the end." So here you see a connection with something that was started in Moscow in the 20s, was brought to the West, came together with the young physicists of the 20th century, and was now read very broadly. So Delbruck left Germany, and he ended up in the United States. In the States, he met with another emigre scientist, Salvador Luria, who came to the US from Italy. And Luria, as it turned out, read the 1935 paper written by Timofey Rysovsky, Zimmer, and Delbruck. And together, they started working on the physical nature of mutations. So Delbruck came to the US on the Rockefeller Fellowship. Initially, he ended up in Morgan's laboratory in Caltech because he knew he was taught by Timofey Rysovsky that fly is a good model organism, but he quickly realized that fly is too complex for the questions that he is asking, and he started working on viruses. He started interacting with Delbruck, and together they provided an answer to the question about the way in which mutations arise. So in a classical Luria-Delbruck experiment that is taught in every undergraduate course in genetics, they asked whether mutations that make bacteria susceptible to infections by bacteriophages, whether mutations are spontaneous or whether they are induced by the presence of phages themselves. And I want you to realize that this is essentially the same question that Четверяков was asking with his students, including Timofey Rysovsky, whether mutations are induced by laboratory procedures or they are present in natural populations, even though in this case, the natural population is just a population of bacteria growing in a flask. So this experiment that showed that in this particular case, mutations arise spontaneously was published in 1943. And it was a stepping stone for the Nobel Prize that was awarded in 1969 to Delbruck, Hershey, and Salvador Luria for their discoveries concerning the replication mechanism and the genetic structure of viruses. Luria's first student was James Watson, and the rest is history, which brings us to the age of DNA. So these are a couple of quotes from Delbruck's Nobel lecture. During the years of 1930 to 1937, while I was assistant to Professor of Lise Meitner in Berlin, and this is the same Lise Meitner who was participating in Otto Hahn's experiments that eventually led to nuclear fission and the creation of atomic bomb, a small group of theoretical physicists held informal private meetings at first devoted to theoretical physics.
Stas,
Yes.
sorry, you have a couple of minutes, like
No
5
problem,
minutes, and
no
we need to
problem.
end. Okay.
At first devoted to theoretical physics, but soon turning to biology. Our principal teacher in this latter area was a geneticist, Timofey Rysovsky, who together with the physicist Carl Zimmer at the time was doing by far the best work in the area of quantitative research, in the area of quantitative mutation research. Discussions of these meetings within our little group strengthened the notion that genes had a kind of stability similar to that of molecules in chemistry. From the hindsight of our present knowledge, one might consider this a trivial statement. What else could genes be but molecules? However, in the mid-30s, this was not a trivial statement. Genes at the time were algebraic units of the science of genetics, and it was anything but clear that these units were molecules analyzable in terms of structural chemistry. So another beneficiary of Koltsov's school was Theodosius Dobzhansky. So here is a quote from Koltsov's report, the one that I already used in the talk. He said that Dobzhansky, so report was written in 1954. So Koltsov is writing about a student of who is 24 years old. So Dobzhansky, who works in Kiev, but in intimate connection with our institute, is studying the structure of genital apparatus in different mutants of Drosophila. These experiments eventually contributed to Dobzhansky's fame that explained the mechanism by which reproductive isolation. And Dobzhansky, of course, is known for the famous quote that nothing in biology makes sense except in the light of evolution. Dobzhansky wrote about Четверяков is that in his classical paper, he showed that evolution is brought about by natural selection acting on a store of genetic variability generated by mutation. Between 30 and 32, the same theory was expounded with greater mathematical refinements by Fisher, Wright, and Haldane that I already quoted, and the four founding fathers, in which he counts Четверяков, thus provided the cornerstone of what later became the biological or synthetic theory of evolution. So his work is not very well known, but as it turned out, it stimulated a great deal of research of activity in Russia and eventually abroad. So where are we now? So we're quite far away from Gelendzhik. So here is an image of a website that contains hundreds or thousands of sequenced Drosophila genomes from different parts of the world. So this resource is called the New Drosophila Evolution Over Space and Time. So instead of looking at morphologically observable traits, we now have genomes of all of these species. And of course, these efforts are not limited to insects such as Drosophila. So we have UK Biobank, in which we have 500,000 of people, the All of Us, which is an American genetics resource, and the FinGen, where populations are surveyed for their genotypic diversity. So this is an example of an address by Bruce Gelb, who was last year a president of American Society of Human Genetics. And he said that because of this flood of information about genetic makeup of individuals and of populations, we need to know how to act on this information. And of course, it turns out that if you look in completely healthy, asymptomatic individuals that you find in places like biobank, you find a lot of alleles that are associated with mutations. So you need to understand what is it that contributes to the ability of a gene to realize its phenotype. And one has to separate, one has to understand phenomena that have to do with penetrance and expressivity, whether or not there will be a phenotype, and if there is a phenotype, then how strong it will be. So it turns out that the terms penetrance, expressivity, specificity were posed 100 years ago. So here is an image from a paper by Timofey Brysovsky in 1927, in which he uses venation of Drosophila wings as a phenotype to examine under which conditions, in which genetic backgrounds, in which environments the gene leads to its manifested phenotype. So in the last couple of minutes, and I have to say that we were delayed a little bit because of the issues with audio, so I hope I can finish it. I really need just 5 minutes. So I want to explain how I, who is not a historian, got interested in all of this. So we were working on a class of human diseases that are associated with paternal age effects. So these are mutations that arise in sperm of aging males. They are not harmful to the carriers of mutation, but when passed to the next generation, they contribute to a large number of diseases, such predisposition to cancer and autism spectrum disorders. A large number of these mutations are associated with mutations in components with highly oncogenic RAS signaling pathway. We took one of the components of this pathway, we took a panel of mutations that was identified in humans, and we introduced them using CRISPR gene editing into Drosophila, trying to understand under which conditions they lead to phenotypes that depend on the signaling pathway. So what we found is that the effects of these mutations, even in flies, where things are much simpler than in humans, and where we can control environment much better than we can in humans, and genetic background much better than we can in humans, the effects were partially penetrant, and the expressivity was different. So we wanted to understand what controls survival, whether it's modifiers, the presence of other genes, whether it's environment, whether it's pure chance. And very quickly, we realized that it's difficult to answer these questions. And we decided that one way to pursue this question is to look at phenotypes that are bilaterally symmetric, where there is a structure on the left and the structure on the right. And we're filled with such structures like ribs and muscles. So we focused on a type of cells in Drosophila that are called tracheal cells. These are like shower heads that deliver oxygen to the muscles in the fly. These structures depend on signaling through the pathway that is mutated in humans and gives rise to cancer and to these developmental abnormalities. And when signaling is increased, instead of one cell on the left and one cell on the right, you generate more. We discovered that this is indeed the case. So under normal conditions, there is a cell on the left, like this green one here, and the cell on the right. When signaling is increased, we see 4 cells in some cases. But we also discovered that the phenotypes are asymmetric. So in the same animal, where the genotype on the left and on the right is the same, and when in the environment is the same, the effect can be stochastic. So in addition to the difference in environments, and in addition to the difference in the genotype, there can be also chance. So we demonstrated using relatively simple statistical methods that unambiguously the phenotypes are stochastic. We were extremely proud of this result, both me and my co-authors, students and postdoc. And then we realized that we were scooped 100 years ago. And we were scooped by a person who was 24 years old or 23 years old, who was one of the members of the Четвериков's team. He discovered using much simpler techniques, much cruder tools, also asymmetric phenotypes, and he did essentially the same statistical analysis to convince himself and other members of the scientific community that chance is at play in manifestation of the genotype to phenotype. So again, the paper was published in Russian 100 years ago, and for some reason it was not very well cited. It was not on my radar. So these are the conclusions. So the work of Четвериков, that is turning 100 years next year, launched the field of experimental population genetics featuring a very clever blend of experiments and theory. Even though the work of this group was abruptly interrupted in 1929 when Chitvirikov was arrested and exiled, it continued through the efforts of his disciples. In the West, most scientists know his work secondhand at best. Most people are familiar with these ideas from the book by Schrödinger, who knows about it from the paper by Timofeyev, Delbruck, and Zimmer that eventually goes back to Moscow. The papers of his school, as I found firsthand, contain a treasure trove of ideas for a large number of ongoing studies in both fundamental and applied genetics. And I would like to end this by a quote from Mikhail Bulgakov, a Russian scientist who was also harassed by Stalin and his henchmen. So this is a quote of his novel, from his novel Master and Margarita, that in Russian reads "Рукописи не горят," which the literal translation is "manuscripts do not burn," but it also means that you cannot kill an idea. And even if the paper is published at the time of the oppressive regime and in a place with limited resources to do science, eventually these ideas permeate and become available to the rest of the community. Thank you very much. I'll be happy to stay on for a little bit and answer questions.
Question? Ute?
Yeah. Stas, can you hear me?
Yes.
Okay. No, thank you. I really, really liked your talk, and not only because you mentioned Löw, but I want to know whether you characterize Chetverikov as a revolutionary who stands on the shoulder of giants, like Mendel, or did this not? Is this not the proper characterization?
I don't think he was revolutionary, but I think he was. I don't think he was. I don't think he was a revolutionary, but I think he was extremely original and forward-looking. And he, even though he did not publish very much, so he established a school of thought that was extremely stable and extremely impactful. So I also have to tell you, Uta, that as someone who grew up in the Soviet Union, I do not like the word revolutionary too much. And I'm not really joking because with me, this word is associated with extreme violence. And I think people like Boveri were indeed revolutionaries, or people like Kaltsov were doing something extremely, something that was never done. So Chitvirikov was an extremely deep thinker, and he was made of the stuff in which scientific progress is made. So he was not Mendel, but he was what he did was absolutely essential, and it was so simple that it is absolutely remarkable that in decades of arguments that mutants have absolutely nothing to do with the progress of evolution, it's remarkable that nobody thought of doing this experiment, essentially doing crosses between the progeny.
Thank you.
So
I understand you, but anyhow.
I think Timofey
thanks.
I think that Timofey Rysovsky was indeed a revolutionary.
I don't think so.
Yeah. Okay. We can discuss when I'm there in.
You can discuss this later.
Because I wrote about him, and I don't think he was.
Okay, let's thanks Stas again.